Thank you very much, Fran. Thank you very much for the introduction, and thank you to both you and Phil and the rest of the focus group for the invitation. It gives me great pleasure to talk to you this afternoon and give you a really small insight into nanomedicines to enable innovative medicines, a very kind of basic view about how I think or a short view, should I say, about how we think they can be applied in a couple of examples, and then we'll move into a more of a fireside chat, I think it's been called, which makes me laugh because it's close to midsummer. So, nanomedicines. I don't think for this audience I need really any introduction, but it is a unique size, shape, composition that really affects the distribution of nanomedicines and means that we can have a number of applications. I like to think of them in three main buckets. Those that are majority of the drug itself, those where the drug is chemically linked to a carrier, and it's that linkage that is important in it, encapsulating the API to the delivery system, that nanocarrier, and that's either antibody drug conjugates or polymer drug conjugates. And those where, as really the drug is encapsulated in the carrier, either a lipidic, a peptide, a polymer, or combination to make a number of architectures, including liposomes, polymeric nanoparticles, lipid nanoparticles, polymerisomes, or the inorganic particles. So there's really a wealth of different types, and just showing here an extract from a nanotechnology report from the FDA, just showing that the number of product submissions to the FDA containing nanomaterials, and it's really growing year on year, and I think this stops in 2019 and is continued to grow and still on the rise. So where are the applications of nanomedicines? How are we going to enable those innovative medicines? So many drugs at the minute are small molecules. You know, still 79% of all applications last year to the FDA were small molecule based drugs. And I think within the small molecule world, there are a number of important applications. On the left, we have poorly soluble compounds. So still 64%, it's estimated, of the compounds in development in the pipeline are poorly soluble. We know that by reducing particle size, we can improve oral bioavailability, and then there's nanocrystals can be used and have been used in that application. We can also use them for intravenous delivery or subcutaneous delivery routes. We can also use nanocrystals for long-acting injectables to improve patient compliance. And you know, the problem with many long-acting injectables is getting enough of the dose in the small volume. And if you can work with the nanocrystal itself and have really high drug loadings, you know, 80–90% of your formulation drug loading, that's going to help with longer term dosage forms. That's really important in infectious diseases, particularly where we can minimize visits to hospitals. Another key area I've really worked on is trying to improve the therapeutic index and use nanomedicines to change the biodistribution of drug to improve therapeutic index. And that's because still the biggest reason for attrition is that lack of safety efficacy balance. You know, we reckon it's about 70% of drugs failing during development due to lack of therapeutic index. And the fourth reason for small molecules is really to enable them to cross biological barriers, either through the gut or the blood-brain barrier, by formulating them within a nanocarrier. However, small molecules can only be used for a selection of diseases. As we understand more diseases and we open up drug targeting space, there are a whole plethora of next generation therapeutics that will be used as drugs as the future. And in this slide, and you know, I love this slide.
Marianne, I think you've been accidentally muted or...
Yes, I saw that. Can you still hear me? And was I on this slide?
You were on this slide.
I think I was muted by a host, but hopefully I'm okay. Apologies. I'll continue. So I'm not sure where I lost everyone, but just to really say that in terms of drugs of the future, I think we'll move out of the small molecule and antibody space and have a plethora of different modalities that will allow us to prosecute different targets, those so-called undruggable targets. And those modalities on the bottom row will be peptides, or particularly the nucleotide-based therapeutics will need nanomedicines to ensure intracellular delivery. So I guess that the challenges here for many of these new modalities is getting into the cell. Just, I'm sure many people are aware here, but the sizes are far bigger than small molecules, 4,000 Daltons going up to 10,000 Daltons for some self-replicating mRNA. They're highly charged. They need condensing to get through that phospholipid cell membrane. And then when they get into the cell, they've got to get to the right part of the cell to either knock down a protein or make it be translated into protein to be disease modifying. And as we come into the cell, it's the key bit. It's relatively easy to get into the cell and the cell uptake. The real problem is what happens within that vesicle, within the cell, and how we get out of that cell. It's roughly 1 to 2, maybe 5% of material that gets into an endosome comes out. And it's really that's, nanomedicines really need to crack that barrier. And given these are some of our most expensive medicines, having such low bioavailability is a real challenge. So in terms of nanomedicines and designing nanomedicines, I can't emphasize enough that the importance is a disease-driven design, really understanding the interaction between the tissue, the disease tissue, and our pathophysiology of disease, the drug and its target, and the delivery system, and the integration of those three in the design of any nanomedicine. So we've got experience, or I've at AstraZeneca with myself and my colleagues in 4 main nanocarrier platforms. We have the polymeric nanoparticles, PEG nanoparticles, polylactic acid, pegylated polylactic acid, where the PEG is used as is that stealth layer. In this nanocarrier, the drug is encapsulated, so we can get about 10 to the 4 drug molecules per nanoparticle, and the size is roughly 70 to 150 nanometers. We've also worked with a lot smaller nanoparticles in terms of the dendrimer technology. So the dendrimers are polymers that are branch polymers that are built up layer on layer. We tend to work with a 5th generation dendrimer. That's most of our experience with to date. That gives us 64 functional units. We can put drugs on 32 of those, giving you 32 drugs per nanoparticle. And then the rest of the surface is for a stealth polymer to disguise the nanoparticle from the immune system. And they're a lot smaller, roughly around 10 nanometers. So about the size of many antibodies within the body, which helps with tissue penetration. We're also working with some internally with some telodendroid mice cells, which we can both conjugate and encapsulate drugs from similar materials. So we're using a lysine core and a sarcosine outer layer much of the time, and we can tune those size from 25 to about 150 nanometers. And then on the right here, or probably we've got our lipid nanoparticles. So I'm sure many people have heard about LNPs. They've certainly been used for the vaccines. This is where both nucleic acids tend to be encapsulated. It's a charge-charge interaction in the core of the nanoparticle, and we can tune those and make those to various sizes. And I think when we've worked with them, certainly internally, we've been really thinking about that design from concept to make sure we can get and make translation to the clinic and to commercial. So I'd just like to illustrate very, very quickly three examples to show you what we're doing. So we have our Aurora kinase B, AZD2811, in the PLA PEG nanoparticle with a really slow release rate. This is a selective Aurora kinase B inhibitor. Aurora kinase B is really important in cell and antibiotics and in cell cycle. What we've managed to show was that when we formulated the Aurora kinase in a nanoparticle, it was carefully designed for the right release rate. In the bottom, this tumor growth inhibition graph, we have really durable tumor growth inhibition at half the dose intensity of the free drug, which is shown at the top. And that was a drug that had shown a proof of concept in phase 2. So we've now been able to show some durable clinical benefit and encouraging overall survival in second-line setting in small cell lung cancer patients, and we're now doing further phase 2 studies in combination with durvalumab. So the second nanoparticle which put into a clinic is a dendrimer. This was in collaboration with Star Pharma, a drug delivery company based in Australia. And what we did here was conjugate a dual BCL2 BCL-XL inhibitor to the dendrimer core. These are challenging molecules because they're BCL2 BCL-XL. It's a protein-protein interaction, so it's quite a large molecule. It's a very lipophilic molecule, but we had to stop development due to the cardiovascular tolerability. So by conjugating it onto the dendrimer, so 32 molecules onto this polylysine core with a PEG stealth layer, and the drug was linked through a hydrolytic bond, we managed to dose a really poorly soluble compound and have a circulation time that was which is prolonged enough to enable us to change the therapeutic index. And I'll just show a little bit of illustration here. So on the right, we can show that once we did mathematical modeling, which is described in the paper referenced here, to aid the design of the release rate. And once we got to the right release rate, we can show on the left that we've got nice tumor growth inhibition in this mouse xenograft model, and on the right that we dealt with the cardiovascular tox. So if you look at the red line, that shows us the QRS amplitude following dosing in a rat model, and some of the initial conjugates we show in the blue line show we still had a depletion in that QRS. It was a lot better, but what we did was use this mathematical model to find that the right release rate to actually minimize completely this QRS signal, which is showed in the yellow, very similar to the control in the black, but we still managed to maintain the efficacy. And that has enabled us to take this drug into phase 1 clinical trials, and we think we've estimated in both in rat and dog a roughly 20-fold improvement of therapeutic index. And BCL2 BCL-XL inhibitors are thought to be important in a number of cancers, and particularly in hematological cancers. And a third and final example I just wanted to share with you was this on an mRNA LNP lipid nanoparticle, where we've worked with Moderna on an IL-12 program. So IL-12 promotes anti-tumor immunity, but yet systemically it's really toxic. So what we've managed is to make an mRNA that codes for IL-12, put it into an lipid nanoparticle, and this has been developed as a novel intratumoral based therapy, and that is now in early clinical trials. So hopefully I've shown you here just 3 examples, 3 applications, small molecules, 2 small molecules struggling with therapeutic index, and an mRNA example where we've used nanomedicines to manage to progress from preclinical development into clinical development. And I think what we've done and what's really important in nanomedicine design and development is to make sure, as well as having the delivery technologies, we've got the capability in place to understand the in vitro characterization, biodistribution through both modeling and imaging, understand cell type and tissue type localization, and particularly those that need intracellular delivery, receptor cell binding, internalization, and cell trafficking, and understand where things go within the cell. I was going to try and summarize this very quick kind of insight into, my view of the progress in the design of nanomedicines. So I think many of the first generation nanomedicines, the liposomes are on the market, and they show they can improve therapeutic index, but probably mainly through improving tolerability. I think some of the first generation polymer conjugates had 2 shorter half-lives, and some of the first polymeric micelles had very low stability, and there was limited tunable release, and cytotoxics were the primary carrier. I think now we've moved to perhaps smaller nanoparticles. We've managed to prolong those circulation times, and that's allow us to up the injected dose per gram in tumor, or up the ratio between tumor AUC and plasma AUC. By controlling the release and understanding the release from the nanocarrier, I think that we can further optimize therapeutic index. Using molecular targeted drugs, you've got then a combination of medicinal chemistry and drug delivery to improve therapeutic index, and getting higher drug loading, I think, has meant we've had less excipient load to patients, which is important. I think the 3rd generation, and it's coming, will be the intracellular delivery, and we're seeing both, targeting by ligand targeting approaches. We'll see greater site specificity by using pH, enzymes, redox. We'll see more combination nanos. I think there's one on the market, Bioxis, at the moment, but we'll see more of those. And we'll see more nanomedicines moving outside oncology. Most of the examples that we're aware of are in oncology. And I think the 4th generation will be exploiting perhaps the immune system, lymphatic delivery, spleen targeting, different routes of administration, and some of the myeloid targeting systems, which are certainly coming through in the literature. So I think that's my little overview of the design and where some future applications may be. I just wanted to really end on the pharmaceutical development. I think it's really important that we don't design things that are so complex that we can't then develop them. We need to make sure that we've got advanced analytical tools to really determine those critical quality attributes that will affect in vivo performance, and we can guarantee them each time. We need to make sure that we've got processes that are sustainable and are robust, and it's really the process that will ensure the quality of the product. So we really must need to understand that. And then that we've got the regulatory discussions and advocacy to make sure we can do that clinical translation. And before I end, I'd just like to say I've given you some of my thoughts about where nanomedicines can be used to enable next generation therapeutics. But I'd like to acknowledge, from the work that I've given you a glimpse of here, my many colleagues that worked in AstraZeneca in the areas of nanomedicines, polymeric therapeutics, intracellular delivery, across a number of departments, my own department, pharmaceutical sciences, and other departments within the company, and the project teams for some of the data I've given you a bit of a glimpse of, as well as our many collaborators, particularly those from X-Bind Therapeutics and Star Pharma. And thank you very much for your attention, and I'm happy to take any questions.